Hi, I'm Elliot. I'm a postdoctoral fellow in Sangeeta Bhatia's lab here at the Massachusetts Institute of Technology. Today we're going to prepare cell cultures on silicon micromechanical chips. The devices look like little combs that lock together, and they allow us to precisely manipulate the interactions between two different populations of cells. Today we're going to study the interaction between liver hepatocytes and supportive stromal cells, specifically 3T3 fibroblasts. Cells are cultured on the top surface of the combs so that by shifting the comb fingers, you can move the cells and change their spatial arrangement. Each device is divided into two parts, which snap together in two possible configurations. The first brings the two cell populations into contact with each other. The second separates the two populations slightly so that the cells cannot touch. Here, contact interactions are prevented, but interactions through secreted factors are preserved. Let's start by talking about how to handle the parts with tweezers. I like to use both larger Teflon tweezers as well as smaller round-tip metal tweezers. With the larger tweezers, you can pick up the parts on the edges like this. With the bigger parts, with the arms, you have to be careful not to break the arms because they're very fragile. So you want to pick them up at the back of the part like this. With the metal tweezers, you can reach into the hole and pick up the chips directly. The larger parts